Hello friends, now in this 7th lecture today, we shall take up another very important process relating to protein foods. We will try to understand the chemistry of processes like denaturation, foaming and gelation of protein foods and see how these processes influence the characteristics and properties of the food materials in different operations. Proteins are macromolecules of great size and complexity. They are of colloidal dimensions and as such they cannot pass through semi-permeable membrane. They are amphoteric in nature, that is they behave both as an acid and as a base. In their polymeric forms, they possess a regular, specific folded 3-dimensional conformation. About 20 amino acids are coded genetically which make up food proteins. These amino acids are linked covalently by alpha carboxyl group of one amino acid and alpha amino group of the other amino acid through peptide linkage. Food proteins are important to texture, color, flavor and functional properties of food. Proteins function as buffering agents, emulsifiers and fat mimetic agents. Now, let us see what are the structures of a protein. When we talk about structure, we normally take this in 4 labels. Primary structure, wherein we understand the linear sequence of amino acids, like you can see here in this, that is how these amino acids are sequenced, and mind it, even the specific position of the amino acid is very important. So, number of amino acids and linear sequence is taken in the primary structure. Secondary structure of a protein involves regular structural patterns such as alpha helical or beta sheet configurations which a protein might take up. In the tertiary structure, 3D arrangements of the atoms in the polypeptide chains are understood. That is how a specific random coiling takes place and then folding in the secondary and tertiary structures. So, these 3 primary, secondary and tertiary structures normally deal with about a single polypeptide chain which are called subunits. When, and it may so happen that in a protein there might be more than one subunits. So, how these different subunits interact among each other to give the final shape of the protein molecule are understood in quaternary structure. When these food proteins come across different agents, when they come across different conditions during processing and handling, there are certain changes take place in the characteristics, in the properties of the protein, that there are certain structural changes take place into the protein, and this we generally call as denaturation. So, the denaturation of a protein may be any non-proteolytic modification of the unique structure of a native protein, giving rise to definite changes in its chemical, physical or biological properties. Mind it, there are no proteolytic decomposition, no proteolysis, there is no breakage of that peptide linkage during denaturation process. It may be unfolding, uncoiling of the organized protein structure. That is, you can see here in the picture, that is the normal protein structure. If it looks like this, then during denaturation process, then organized structure is opened or unfolded. In some cases, it may again renature to get the structure. So, actually in the denaturation there is loss of secondary, tertiary and quaternary structure of protein, but definitely not the primary structure. As I indicated that there are various factors which influence the denaturation process. These factors may be the physical factors like temperature, high pressure, irradiation, sound waves and shear forces etc., to which the protein come across during processing or handling. The chemical factors like pH, polarity of the solvents or additives, even the biological factors such as enzymes are those which influence the rate of denaturation processes in protein foods. There may be two types of denaturation reactions or processes depending upon the factors, depending upon the food characteristics, depending upon the severity of the process to which these food proteins are exposed to, and that may be a reversible denaturation or irreversible denaturation. In the reversible denaturation, the effect of denaturation reagent, if they are removed by processes like dialysis etc., then the protein may again renature. But in the case of irreversible denaturation, there is formation of an insoluble substance called generally as coagulum, as happens you can see when the egg is boiled. So, in the heating of egg white above certain temperature, maybe 50-60 degrees Celsius, the coagulation of protein takes place and then this becomes some sort of irreversible denaturation process. The different changes that take place in protein characteristics, in protein properties during denaturation include decrease in its solubility, there may be loss of biological activity of the protein, it might not be possible to crystallize the protein once it is denatured, there might be increased reactivity of constituent groups, there may be a change in the molecular shape of the protein or it may become more susceptible to enzymatic hydrolysis. Denaturation often improves the functionality of the protein like digestibility. It also inactivates sometimes the anti-nutritional factors. It might inactivate deteriorative enzymes and it may also provide method to monitor processing of food. Now, let us see that what are the different functional characteristics of the food, protein, how it behaves, the functionality of the food. The function of protein is to provide and stabilize the characteristic structure of an individual food. The ability to form and or stabilize networks, that is gels, films, foams and emulsions etc., or the salts are described as functional properties of the protein. Various characteristics that govern the protein functionality include shape of the protein, its size, amino acid composition and sequence of the amino acid, how the protein interacts with other components, how is the molecular flexibility or rigidity, its secondary, tertiary or quaternary structure, even the net charge on the protein molecule or the distribution of the protein molecule or even hydrophobicity ratio of the protein. So, these are the characteristics and properties of the protein which influence its functional properties and therefore, which give sometimes desirable characteristics or undesirable characteristics into the food materials. In this, I have tried to give you a description, that is how the functional properties of the food proteins are understood, what are the different criteria. Like in the kinesthetic properties, we try to find out or see the texture, mouthfeel, smoothness, grittiness or turbidity. This binding property of a protein, we take a functional property, how it binds with the flavoring molecules etc., even the structural characteristics of properties, the elasticity, grittiness, cohesion, chewiness etc. In the rheological properties, viscosity or gelation is understood. Hydration that is an important functional properties of the food. Here solubility, wettability, so water absorption, swelling, thickening, gelling, viscosity, all these characteristics might be influenced by the hydration characteristics of a protein. Even the enzymatic properties, that is there might be coagulation, like in the cheese making, the rennet enzyme, it coagulates the protein. There may be certain enzymes which may cause tenderization of the protein. Example, you can take pepin, that is the pepin enzyme, it causes the softening of the hard tissues of the meat, that is meat become tender, there might be mellowing etc. So, these are the different functional characteristics or functional properties of the protein and food proteins, and how their functionalities, what are the different criteria to decide their functionalities. Further, you can see that what are the different mechanisms which decide a food function, and we take a few examples, like in the beverage, if we take a food, that hydrophilicity of the protein, like meat in the case of milk etc., whey protein etc., how hydrophilicity characteristics will ultimately influence its solubility in a liquid food. Similarly, the water absorption characteristics of the protein may be influenced by its ability to bind with water, that is to form hydrogen bonds with water, and this happens generally in the case of meat, sausages, cakes, breads etc., all right, where this protein, different proteins, they bind with or in the form of with the hydrogen bonds etc., and give the desired characteristics. Similarly, the other process like gelation, emulsification, foaming, elasticity, you see that what are the different mechanism. In the case of gelation, it might be the water entrapment and immobilization of the which might ultimately result to the network formation. Emulsification, the mechanism might be adsorption and film formation at the interfaces. Elasticity may be because of the hydrophobic bonding or even formation of disulfide cross linkages. So, this explains briefly, that is how these different functional characteristics of the proteins are influenced. Now, let us take another important property, that is the foam, how protein characteristics. In the earlier slide you have seen that is foaming and gelling are the important characteristics imparted by the proteins into the food system. So, we can define a foam as a two-phase system consisting of air cells separated by a continuous liquid layer, which is generally called the laminar phase. Small air bubbles imparts body, smoothness and lightness to milkshakes, whipped cream, ice cream, cake, snacks and bread, etc. And there are two important characteristics of the foam. One is whippability, other is foamability. That is, when foam is obtained by a high blending or whipping treatment, that is, if we just give that by some mechanical means we whip or we blend that protein system, some foam is generated. A good example is that egg yolk, that is when we provide. Another is the foamability, that is foam is prepared by injecting air or gas through the protein solution. So, it results into the foam. So, this foamability and whippability are the two important characteristics which govern the formation of foam into the food materials. Foaming properties of the food proteins are influenced by source of protein, method and parameters of processing like temperature, pH, etc., concentration of protein, its mixing time and the method of foaming, whether it is whipping or whether it is other method. The main basic functions of protein in foams are to decrease interfacial tension, to increase viscous and elastic properties of the lipid phase, as well as to form strong films. If we see that how the foam formation takes place, generally the soluble globular proteins diffuse to the air water interface, they concentrate and reduce the surface tension. Proteins unfold at the interface and hydrophilic as well as hydrophobic groups reorient. Even the polypeptides with possible partial denaturation form a stabilizing film around the bubbles which promote the foam formation. Protein foamability is correlated with its capacity to decrease surface tension at the air liquid interface. When the continuous film is formed through the protein interactions as well as through electrostatic and hydrostatic interaction or even hydrogen bond formations also influence to some extent the film formation characteristics of a protein. The foams, once they are formed, in many food processing operations, we desire that this foam should be stable, it should not collapse, like in an important process is the foam made drying, whereby foaming process we increase the surface area and the liquid foods are put into the dryer in a thin sheet of the foam. So, it reduces the drying time, but the important consideration or characteristics of the foam should be that it should not collapse during the drying process. So, the stability of a foam is a very important characteristics. Stability is a measure of the ability of the product to keep a foam once formed and prevent it from collapsing. Okay. And there might be various reasons for the collapse of the foam. It may be because the large gas bubbles grow at the expense of smaller bubbles. The stability of the foam may also depend on the strength of the protein film and permeability of gases, that is how this film is permeable or impermeable to different gases. Even proteins absorb at the gas liquid interface and form a protective film around the bubbles. Foaming capacity is a measure of the ability of the protein to form a gas filled cellular system under specified conditions. So, these two are important characteristics we should see, that is the foaming capacity of a system, that is the foam formation characteristics, and then after the foam is formed, its stability. Ideal foam forming and foam stabilizing proteins are characterized by low molecular weight, high surface hydrophobicity, good solubility, a small net charge in terms of pH of the food, and easy denaturability. That is, if a protein is denatured easily, it may be a good source for providing a good foaming ability as well as a good stable foam formation. After having studied the denaturation of the protein, foaming characteristics, let us see the gelling behavior of the proteins, that is food gel. So, a gel can be considered as the structural matrix holding liquid and can be formed spontaneously by swelling at a high protein concentrates. Gels behave as a solid like material as well as they possess many characteristics of a fluid. Specific properties of the gel are due to the presence of a 3D networks of proteins. Textural properties of food gels and juiciness are determined by the gelling capacity of the proteins. Protein gels may be utilized to simulate the textural properties and mouth feel of the lipids in the food system. So, how gelation takes place? Gelation is defined as a protein aggregation phenomena in which polymer and polymer solvent interactions and forces are so balanced that a network of matrix is formed. In a gel, the liquid prevents the 3-dimensional matrix from collapsing and the gel matrix prevents the liquid from flowing away. This matrix immobilizes extremely large amounts of water. The random aggregation in which polymer interactions are favored over polymer solvent interaction may be defined as a protein coagulation. There may be two types of gel formed: polymeric network or aggregated dispersion. Polymeric networks are the gel formed by gelatin and other polysaccharides such as agarose. They are in normally lower polymer concentrations and transparency and texture are generally better of this type of gels. On the other hand, aggregated dispersions are the gel formed by globular proteins after heating and denaturation. Partial denaturation result in thermoplastic character of the gel. These thermoplastic gels do not liquefy when heated, but they can soften. The gel formation, or see what are the mechanism how these gels are formed. They are formed when partially unfolded proteins develop uncoiled polypeptide segment. Interactions at a specific point to form a 3-dimensional class ring network also result into the formation of gel. Partial unfolding of protein with slight change in secondary structure partial unfolding of the native structure can also be related to the action of various factors, like the gelation process is dependent on the formation of a 3-dimensional protein network as a result of protein-protein and protein-solvent interaction. Gel matrix is a interconnected cage like unit structure saturated with continuous solvent like phase. So, these are the processes or mechanisms of gel formation in various food system. The protein gel formation may be of two types, may be heat induced gel or calcium induced gelation. In the heat induced gelation, an initial step involving the unfolding or dissociation of the protein molecules takes place, and aggregation step is there, in which association and aggregation reactions type of heat induced gel structures are formed. It may be thermoset or reversible or thermoplastic or irreversible gels. The calcium induced gelation is easily seen in the food, like when the milk proteins are coagulated, there is calcium pyrocasein phosphate gels resulting from the enzymatic disruption of the casein in the manufacture of cheese. They are characterized by low gel strength and rapid synersis. Different factors that influence the protein gelation processes include the extrinsic factors of the protein, like character of the solvent, temperature to which the proteins are exposed to, pH, ionic strength, divalent cations, and even presence or absence of denaturants. Most of these factors which I have seen, they also cause denaturation. Other macromolecules present in the system, lipids, or even activity of the enzymes. So, these are some of the factors which are considered intrinsic factors and which influence the gel forming ability of the proteins. Other factors, maybe which generally say primary factors, include the composition of the amino acids, because this composition of the amino acids, it may also influence the even the acidity or acidic nature or basic nature of the protein finally. So, that also might influence or has important influence on the gelation characteristics. Sequence of the amino acid residues, total molecular weight, the tertiary and quaternary structure of a protein, its hydrophobicity or the net charge and the charge distribution on the protein, its isoelectric point, the flexibility of the protein molecules, etc., are the other important factors which influence gelation of a food protein. I have so far given you a brief overview that how different processing conditions, different other conditions to which food proteins either in their native form or in their isolated or precipitated forms, they are exposed to various conditions, and then their characteristics, their behaviors get changed, they may get denatured, they may form different gels, they may form different foams, etc., and these denatured proteins, foams, gels, etc., have their different, they influence the functional characteristics of the protein, they improve their usability in various food formulations, food preparations, etc. This, I thank you for your kind attention.